So the basic point is that research universities are the most powerful source of science-based innovation, because here curiosity-driven research leads to transforming practical applications. Anyway, the basic point is I'm going to give you 3 examples of science from universities. They involve different features of university research that are unique. One is interdisciplinary work. A second is power of combining teachers and students on research projects. And the third is international collaboration. Okay, roll it. Many species of birds use color to communicate. Here you have a superb bird of paradise performing a courtship display, watch this, to a prospective mate. His dance shows a showcase of vibrant blue hues on his feathers. Could this be the origins of the happy face logo? Okay, a Yale team of biologists who was curious about how these colors are formed, and an engineer have actually combined, have actually combined in their work to understand what are the physical origins of these vibrant colors. And they found that the vivid blues and the vivid greens that are shown here do not come from pigmentation in the feathers, but rather from the structure of the feathers, the interaction of light with tiny nanostructures inside the feathers. Now, the vivid color of the eastern bluebird, for example, is formed by a chaotic network of air channels that are in the feather protein. Each channel is only 150 nanometers across, and these tiny feathers selectively reflect only blue light. To communicate effectively, each bird must precisely control the size of the air channels, and we're discovering how they do it. Yale researchers have discovered that the protein nanostructures are self-assembled by phase separation, much as gas bubbles form in champagne. This movie shows how the unmixing of 2 fluids can create the chaotic channel structures that are observed in the bluebird. These insights may have important implications for photonics technology. You know, telecommunications relies on the flow of light through fiber optic cables, and standard technology involves cladding the light to keep it from dispersing. Yale researchers are discovering that cladding that mimics the design of bird feathers actually works better than more regular nanostructures. These biomimetic designs have immense potential for novel capabilities. Second example, Professor Scott Strobel leads undergraduates on an annual expedition each year to discover microbial diversity in the world's rainforests. Students design and implement their own experimental plan, and this results in a strong sense of project ownership. During these trips, students collect small samples of plants for analysis in Yale labs. They're looking for endophytes, fungi that live within the tissues of higher plants. They culture the organisms, identify their phylogeny, test the bioactivity of the organisms. And there are about 1 million species of fungi on Earth. Only 5% have been discovered and described. And thus, it's common for students in the course of their studies to discover not just new species, but actually whole lineages, major lineages of organisms. These organisms have the potential to treat disease and sustain life on Earth. For example, on the left side of each panel, you have something isolated from rainforest trees. They produce gases that can kill the bacteria, potentially, on the right of each panel. You see the one on the lower right actually is an effective antibiotic. Not all of the useful products are disease-related. Strobel and his students have discovered endophytic fungi that degrade cellulose and excrete carbon in the form of molecules with the same chemical composition that's found in gasoline and diesel fuels. This is octane and other medium-chain length hydrocarbons. Fungal production of such products is unprecedented, and it has significant potential for providing cleaner fuels. Discoveries such as these could be missed if we don't preserve the world's regions of high biodiversity. Another argument for biodiversity, because it has immense economic potential in the living wild forest. Protecting biodiversity will ensure a long line of exciting innovations. Degrading biodiversity will cut off what might be a bountiful source in the future. Genetic analysis of the mouse, which has 99% of its genes just like humans, could provide the first functional map of the mammalian genome. At present, we only know 15% of the genes in their function. One way to gain this information is to mutate so that each gene that can be identified as to its function, and our Yale scientist Tian Xu is doing this. He's following on the work of Barbara McClintock, who won a Nobel Prize for discovering mobile genetic elements that jump within the genome, transposons. For more than 60 years, we've tried to move from corn to mammals, and with only limited success, but Dr. Xu has found a way to do this. He uses a piggyback transposon. It's very highly efficient and fast, and it allows thousands of mutant mice to be generated. Some you can see have obvious physical defects. We can also generate mutants that can be phenotyped to find diseases that have medical conditions that might, or other medical conditions that might affect humans. Dr. Xu is generating a sort of genome-wide set of mouse mutants, each strain bearing a single mutated gene. Then he phenotypes them through a battery of tests, and the informatics database from this functional map should have significant impact on medical diagnosis and on identifying therapeutic targets. Yale is doing this work in collaboration with Fudan University in Shanghai, where the piggyback mutagenesis is ongoing to generate a library of mutant mice. Labor is cheaper in Shanghai. You can do this very, very efficiently. Then we bring frozen embryos back to Yale, to our new West Campus, where we've established a mouse clinic for the systematic phenotyping of these mutants. In summary, each of these examples, you can see how university can be a powerful source of innovation. And as you saw in turn, the first example involved interdisciplinary collaboration. The second, utilizing the interaction between teachers and students. The third, international collaboration. Thank you.